Hello everyone, I'm Jayoung Ryu and I'm excited to share our work on modeling endogenous editing outcome for base editor reporter screen with CRISPR-Bean. So the question in our mind in genetics is to better understand how sequence variation impacts disease or various human phenotype. And there are three main approaches that we can tackle this shared question. First, association-based method, including GWAS and QTL studies, and prediction-based method, which were very nicely presented by the previous talk, and the perturbation-based method. And among these approaches, perturbation-based method can tackle the limitation of association-based method that we can disentangle the variants with the linkage disequilibrium, and we can also induce a variant that may potentially have too high impact to be observed in significant amount in population because it's just too harmful due to the selection. And among the perturbation method, the CRISPR-based methods have its own advantage that it can induce the sequence variation in the endogenous setting, where it is under the same environment, including epigenetic modification or chromatin conformation. And because we were interested in the single nucleotide variant, we are focusing on CRISPR-based editing technique here, which currently has higher efficiency than prime editing. So I'll briefly touch on how CRISPR-based editors work. In the engineering setting where we are trying to install the variant that we want, we're trying to install the variant that we intend, we are transfecting the guide RNA inside the cell, which will then be integrated into the genome, and it is then transcribed into the guide RNA, which will bind to the base editing enzyme, and will guide this enzyme to the endogenous target site that has the complementary sequence to the guide RNA. And once the base editing enzyme arrives to the endogenous target site, it can induce the single nucleotide mutations. And there are multiple types of base editors, including C base editors, A base editors, and recently developed transversion ABEs can even install the transversion, which now covers 84% of human pathogenic point mutations, suggesting the huge clinical potential of base editing. The main caveat, though, for the base editing is that base editing outcome is very highly variable due to multiple factors, including the target sequence and the target adjacent sequence, what is the enzyme that we're using, or what are the cell type that we are working on. These factors, as in outcome, induces two different outcomes. First, variable editing rate at our target base. So different guide RNA will induce different levels of editing. Second, bystander edits can happen, which means the adjacent bases that we didn't intend to edit can be edited together or edited instead of what we tried to do. So these two outcomes will confound our analysis in identifying the correct causal variant and correct variant effect sizes. So our experimental collaborators tried to overcome this limitation by developing a clever and simple trick. Here, as I briefly touched upon, we originally had a spacer sequence which will be transcribed into guide RNA and will target the endogenous target DNA. Instead of having only this, in the construct that we're transfecting into the cell, we are sticking the reporter sequence, which is basically the copy of the endogenous target site DNA sequence with the flanking surrounding sequence, so that as the outcome when we are reading out the guide RNA information, we are reading out the paired information about what the guide is inside the cell, plus what is the editing outcome that guide induced. So because we can do this in large scale, we can read out the per guide genotypic outcome by just replacing the original construct with the reporter construct. We, in addition, observed interesting observation here. We tried to compare the reporter editing versus the endogenous target site editing in 4 different loci and found that although we observed that reporter editing and endogenous editing are very decently correlated, but while also being scaled by the accessibility of that endogenous target site we're targeting. This makes sense because if the target site is not accessible, it is hard for enzyme to bind there and make the edit. And we will later use this relationship of reporter and endogenous site editing rate ratio will depend on the accessibility in our modeling. So, okay, we now know that we can use the reporter to read out the endogenous editing outcome, and also it will be scaled by accessibility. How can we use this new layer of information to tackle our original question about how to better identify what is the impactful variant for a given phenotype? And I want to provide our study as a demonstration of this base editing reporter framework. So our phenotype of interest is the LDL cholesterol level. LDL cholesterol is the causative phenotype, intermediate phenotype for the cardiovascular disease, which are both common and polygenic. And one additional critical point is that LDL-C level is measurable for type, and we can sort cells based on this phenotype. So we can use this in the large scale screen, which I will further elaborate later on. And it's also studied very extensively in the observational study and the well cataloged in biobanks, just because it's very easy to measure from patients' blood. So from this LDL-C phenotype, we want to ask 2 different questions through the CRISPR screen. First, can we test or validate the GWAS candidate hits? Because the base editing variant can disentangle the LD variants. And second, can we discover novel coding variants of very large gene when we cannot really read the editing outcome directly out? So we are looking at the LDL receptor, and this library of guide are tiling across the more than 5 kilobase pair of exonic regions. And in this library, we are trying to induce every possible missense variant that can be induced by A base editors. I'll first explain about the CRISPR screen before I go into how reporter can be useful here. So in the CRISPR screen, we have a pool of cell, and we're transfecting, because we are trying to test or rank different variants based on how much they impact the phenotype of interest that we're looking at. We are transfecting thousands or more than 10,000 guides that are targeting hundreds to thousands different variants. And assuming that we have one or very few number of guides per cell, we are, after the guide has been integrated, it will be transcribed and we'll be targeting the endogenous target site, which will change our phenotype of interest somehow. And depending on the phenotype, we are selecting or sorting the cells. And based on the sorted phenotype, we can have different population of the cells, and we are quantifying how abundant the guide is by sequencing the construct here. And what is usually done is to do the enrichment of how much this guide is enriched in high phenotype cells versus low phenotype cells. And what can be easily done is to calculate log-fold change and somehow aggregate them into the variant level statistics. What is problematic here is that we only read out the spacer or guide RNA identity, and we don't really read out the endogenous target site editing. So it's very unclear how to aggregate these guide level information into the variant level information that we are actually interested. So it's inherently wishy-washy based on assumption that intended editing outcome is the actual outcome and hope for the best. And we want to do better than this by using the reporter. So we have the same screen design, and after having the construct integrated into our genome, the guide RNA will target both the endogenous target site and the reporter. And as we are reading out the editing outcome from the reporter, now we have these another layer of information of what is actually happening in the endogenous site. And we also developed a software called CRISPR-Bean, which can account for both of this information for better estimation of variant impact. Now I will briefly go over how this method works. On the left, I'm showing the plate diagram for who's more used to this notation. And on the right, I'm explaining in more detail. Here, we are assuming the wild type allele has standard normal phenotype, which will serve as our reference about the variant phenotypic shift. So for each variant, we are assuming a normal distribution of some shifted mean or variance. And based on this variant and wild type allele phenotype, we are constructing the cell level phenotype, which is the mixture of when that cell is edited for the A variant or not edited for A variant. And the weight of this edited component or the editing efficiency are read and scaled by reporter editing outcome and the chromatin accessibility of the target loci. In the downstream, we are going from the per guide editing outcome and doing the in silico sorting of cells, just as how we do in the experiment. And from this proportion of cell that falls into each sorting bin, we are modeling the guide count as the over dispersed multinomial distribution. And because we have the observation about the guide count and the reporter information, here we are inferring back the posterior variant phenotype using Bayesian network inference, variation inference, to get the posterior estimate of what is the variant phenotype based on our data. So this is how we jointly use the reporter editing outcome and the phenotypic outcome, which is the guide abundance in each sorting bins. Now I will be showing some of the results. Here I'm benchmarking the method with all other applicable methods to the sorting screens. And second row or the bean is showing the best performance, especially compared to the simple log fold change based method. And the second and third here in the color are the version of CRISPR-Bean where we used less information than the full information of using both accessibility and reporter. So we also see that when the model includes both the accessibility and reporter information, we are working much better. And we are also validate the individual guide transfection and the phenotypic change following the individual guide transfection and observe the descent correlation. When we actually looked at the top hits of the GWAS candidate variant hit, we find out the top variants are located within or very proximal to a gene that is already known to involved at cholesterol transports, which validates our approach. I'll also go on and show briefly about the LDL receptor coding sequence tiling result, where we try to perturb all the missense variant that can be installed by A base editors. Here I'm overlaying the repeat domains and the scores from the method, and this aligns very well with the conservation score that is shown on the bottom. We also did the classification to classify ClinVar pathogenic variants versus benign variants. And again, our method is shown to have best performance. And lastly, we show one of the unique results that we have is that because our phenotype is the continuous phenotype, which is LDLC level, and we actually have the patient phenotype as a continuous value of what is the concentration of LDLC, we can directly compare those two scores, which is the Z-score from our model versus the patient's LDL cholesterol level. So in the UK Biobank, we have the matched information about which variant, patient with which variant has these level of LDL cholesterol level. And this is the plot that we plot on the x-axis, that patient LDL cholesterol level versus the estimated pathogenicity of the variant. And we see moderate correlation, which is improved by the recently released imputation method that tries to share the information between the same amino acid transition. And when we did the very minimal prediction modeling of constructing linear model of this imputed score plus the conservation score, we achieve decent correlation for the unobserved variable in the base editing screen. So this shows that this first demonstrates how we can compare the continuous human phenotype that is actually observed in patients with the screen setting, and we can shows the potential that we can apply this for predictive modeling based on patients' genetics. In conclusion, base editing reporter provides per guide editing outcomes scaled by loci accessibility, and CRISPR-Bean accurately models this together with the guide abundance to give better estimate of variant effect size. And we also show the clinical potential of this approach. I want to end the talk by thanking all the team co-authors who are working on releasing this work, and also the supervisors, Luca and Richard, and especially want to thank those who worked hard on the tech dev. And here is our GitHub. If you are interested, this will be released. We're working on releasing it soon, so please keep an eye on us, and thank you for listening.
So, questions?
Sorry if I missed it, but so does the model take into account those bystander, like the rate of bystander editing, or just the on-target efficiency?
It also takes account for bystander. I didn't elaborate it further here, but there are two different ways you can run model. First, like ignore bystander because we don't expect like effect from non-coding, like non-candidate. Second, you should account for the bystander, especially if you're working on the coding sequence tiling screen. And here I am allowing each guide to have more than just edited allele, but it allows for multiple alleles per guide and information sharing. If that same allele is occurring multiple times between neighboring guide, we are allowing for information sharing.
Oh, I see. So the next target along, you would
Yes.
share.
Yes.
Okay.